Okay, so in today's video, we are going to discuss factors that affect enzyme activity. Factors, factors that affect enzyme activity. So, why enzymes are very sensitive? Very sensitive. Why they're very sensitive? Just because they're made up of proteins and they're sensitive, so they need very specific like temperature, pH, like they need everything should be specific for them. Not every enzyme can work with the same pH, same temperature. They need to be like they're unique in all. They all need their own temperature, pH, these all. So this means they're specific in nature. They are specific in nature. So this means they need particular pH, particular temperature, and like they need everything particular to work properly. Enzyme cannot work on the like pH we are just giving them randomly. They need a specific pH temperature, and that effect can be any like positive or negative both. And yes, G. So if we just given them 10 Celsius, this means their concentration, their ability, like their activity will enhance, and that will come to the double of what they exactly can do on the half, but this would be double. Okay, and if it's double, so this can also be denature, like this means this will not work. Why? Like as I just told you, these are very sensitive, very sensitive. Okay, because they're made of protein in nature, and for that they need optimum pH, optimum temperature. Optimum simply means like particular or specifically, like they cannot enhance or they cannot decrease. If it would happen, if the activity have been enhanced or like increase or decrease, this means they have lose their ability, or they will not work properly. They will be denaturized in their in their environment and so on. So we have, oh my god, where is this arrow? Okay, so first thing that affect this is the temperature. Okay, so temperature, like everything, this was 10 Celsius is all is example of temperature. For every 10 Celsius increase in temperature, this means there is increase in enzymes activity. Okay, as I just told you this before, like temperature, they need very specific temperature. So if I just made this graph, so if this middle, like this one point is the optimum temperature. Okay, and this here, this line represent enzyme activity. This is enzyme activity, I'm sure you will not get my writing. So in this one, like this origin point, they will be inactive. Okay, inactive, and they start working on this optimum temperature. They got this is optimum temperature. They start working, like this was a specific point which they were supposed to get to work efficiently or to work properly. Okay, here these they were inactive. Here they got the optimum temperature. This was the enzyme activity. And now what will happen on this one step, if we just enhance or if we just decrease, so here it will be denaturation, just because they will not work now. So this was, let me just write it. So on this one they were inactive. On the second one they just became denaturation. Okay, then like enzymes, not enzyme, but they need a very like particular temperature. For suppose if you just take example of human are supposed to get 37 Celsius, or like a little change could be 38, but not most probably like more than that or less than that, just because they will not work. And we have a lot of bacteria as well, bacteria.